here's some data, right? And first of all, is this standard error or is this actually error from the data? What do you
What's the
think?
difference?
Okay, so you know how you can say that data points or data within a point is evenly distributed, like it'd be normally distributed, and in that first standard deviation, you got like 68% of the data points, and then it's
Oh
like
yeah.
78 or something for second standard second. So what'll happen is sometimes what they'll do, let's say in this graph in 2002, I took 5,000 samples, right?
Mm-hmm.
And in those 5,000 samples, the average looks like it was around 14 degrees C, right? Right
Right.
there. But the
Yes.
range was way down here to like 2 degrees C and 26 maybe. Okay,
Okay.
so
Okay.
if all of these bars are about the same height, this one's really short, there's none there, then probably they're using one standard deviation from the mean of the data to put the error in. They call that standard error. All right, SE. Now, if it's actually the standard deviation on each side of this mean of the real data, then you start seeing these bars as bigger, right? So this is probably true error and range of data. So a couple things to learn from this. If I look at the 2006 and 2007, what do you notice about that data versus, whoops, sorry, didn't mean to do that, versus all the other data years?
It's smaller, like the ones
Yeah,
in.
the data range is much, much smaller, right?
Mm-hmm.
Now, that could be because there was less variance, but it could also be that maybe in those years we had much more accurate readings. You see what I'm saying?
Yes.
Okay. If all my data is tight, like those two data points, I probably have some pretty good precision in my measurements. If they're
Mm-hmm.
all like 2002, I'm probably not taking measurements very well. So the error bars can indicate when you might have problems collecting data, right? So let's
Yes.
take a look at, let me find another one that we can play with a little bit. I want to find one that's got some a little bit less overlap. Okay, this one could probably be good. Okay, let me pop this one over there. Okay, see if I can make that a little bit bigger. Maybe not. Okay, so here's some data points. And let's just assume that we're looking at the red data points, full sun. All
Mm-hmm.
right. And the question on the AP exam is, okay, I got red maple, sugar maple, and black maple, and I'm looking at diameters in full sun. And I'm asking you the question, is in full sun, is there any difference between the sugar maple and the black maple?
The sugar maple has like a bigger range of numbers.
Okay, so first thing you got to know when they're asking that on an AP test, they're asking you, hey, is there any difference in the average between the sugar maple and the black maple?
Oh, so like the black maple has a smaller average than the sugar maple?
That's what you thinking, but remember what this error bar means here is that really here's the average, but really it's somewhere between here and here. And on this red maple, it's somewhere between here and here. So if these two ranges overlap, there's no significant difference. Okay.
But where do you draw the line between like overlap and not overlap?
If they're overlapped at all with error bars, then
So what
there
if
is
like the
no
error bar
significant
barely,
difference.
what if the error bar barely overlaps? Like it just barely touches each other.
And that's just it. If there's any overlap at all,
Okay.
and there's no significant difference. Now, how about between the red maple and these two guys?
Oh, so then the red maple is smaller than the two.
Right. So let me see if I can find here. So, the body, because we're a complex organism of organs and organ systems, we have to regulate. Okay. And so we call that homeostasis, right? And we have set points for, as you saw in the video, things like sugar level in the blood or our temperature, or I mean, a whole range of things that have to have set points, and the body has to have some way of sensing that and then reacting and regulating it to keep it within a optimal range for whatever it is that it's monitoring. Okay, so we have those normal ranges, and we sometimes call that optimal ranges, right? Because our temperature's never always right at whatever the temperature, 98.6. So it does go up and down. Okay. All right. So the example we'd have here would be body temperature, 98.6, right? But if somebody's down at 97 to 99, they're probably still okay. It's when you go above or below those that you start to wonder, is there something out of whack? Are they sick? Do they have an endocrine regulation problem? And so, you're between 97 and 99, the doctor's office isn't going to worry too much.
Uh,
Okay.
do we need to know the number for body temperature?
No, not usually. Uh,
Okay.
yeah, and usually if it's something related to that, they will tell you what it is. Okay.
Okay.
All right. So homeostasis is this idea of balancing and keeping things within an optimal range. But we have to think about how does our body detect and respond? And that's going to relate to the assignment we have at the end of this. All right. And the way
Mhm.
it does that is it uses feedback loops in order to do that. So if we just sort of take apart the word feedback, what does that sort of mean to you, Leo?
Um, feedback is like getting information back
Yeah,
to
getting
you.
information. Yeah, good. That's probably a really good way to put it, Leo. That you're getting information, but then you're also doing something with the information, right?
You're like using the information to usually, I guess, improve.
Yeah, to improve or maintain.
Yeah.
Exactly. Yep. Good. All right. So there's basically two types, negative and positive, and you need to know some terms that are sort of associated with that. Um, and so these are sort of the four terms we're going to take the rest of this lesson into. Okay. A stimulus is just the variable that causes a response. And then
Mhm.
receptor, sensor, that's sort of self explanatory. It might be the brain, it might be another nerve cell near the brain. Okay. Or that sends messages to the brain. And then
Mhm.
effector, in this case, we're thinking about muscles or glands that will respond to that stimulus and that stimulus that was sensed. And then the response either increases or decreases the effect of that stimulus, right? Um, and so let's get into the two examples that we're going to use for negative and positive. All right. So first thing to know is negative feedback is sort of nature's choice. It's the one that um, is more common. And what it does is it reduces the effect of the stimulus. In other words, it's there to get rid of the stimulus. So things like sweat, blood sugar, respiratory rate, right? I mean, I sweat because my body is sensing that I'm too hot and I need to cool off. And the sweat gets rid of the stimulus because it does cool my body off. For blood sugar, I want to be able to take the sugar that I've gotten from my digestive system from eating, and I want to get it into the cells in order, to do glycolysis and cellular respiration. But if it goes too high in my blood, then it can cause organ damage and nerve damage, and we don't want that, right? So it's something that I want to bring down, but sort of the caution on blood sugar is we don't want it to go to zero, right? So it's a negative feedback loop, but it can also sort of be positive in that I can cause my body to release sugar into the system as well from the liver and the muscles. So that one's a little bit sort of a dual um, example of negative and positive feedback. And then respiratory rate, you know, um, if I'm running along and I'm trying to breathe normally, at some point I go anaerobic and I have to breathe a little bit deeper and a little faster to get more oxygen so that I can do cellular respiration instead of lactic acid fermentation in my muscles, right? So I can sort of control my breathing rate, but there's a certain point that if I exercise hard enough, yeah, I won't be able to control that.
Wait, I don't get how breathing rate is negative feedback. Like, what are you
Well, think
maintaining?
about this. The feedback would be I'm getting the acidity is going up in my legs in the muscles,
Mm-hmm.
and the oxygen level is going down, and CO2 level is going up. And what's
Right.
happening there is I'm not getting enough oxygen to my leg muscles. And so they are doing lactic acid fermentation. Remember, that only gives us 2 ATP per glucose instead of, your normal 32 to 38 for cellular respiration. So there's sensors in the muscles that say, hey, the pH here is going down, CO2 levels are going up. I think we're going anaerobic. And what that does then is it tells your body to start breathing faster, and your heart rate goes up at the same time to get that additional oxygen to your legs that need the oxygen. And that's nothing that you control. It's controlled by sensors within your muscles and your blood system that then control rate. And what's that going to do then? It's going to bring the acidity back up, bring oxygen there. So it's going to reduce the CO2. So the negative feedback is I'm reducing the effects to where I'm no longer doing lactic acid fermentation. I'm doing aerobic respiration. Does that make sense?
Yes.
Yeah, I know it's complex. And this is why I say covering things like the nervous system, the immune system, and looking at endocrine system are probably pretty good. Looking at things like bones or naming all the muscles, probably not going to help you in AP Bio. All right.
Mm-hmm.
All right. So let's take a look at an example of body temperature regulation. So you probably recognize this guy, right?
Patrick? Yeah.
Yeah. Okay. So the stimulus we'll say is going to be heat. The receptor is temperature receptors on the skin. The effectors would be the sweat glands. And the response would be sweat. So
Mm-hmm.
then you end up, of course, with sweaty Patrick, right? Now, let's take a look at the other side. What if the stimulus was cold? What would the receptor then be? Or where would the receptors be?
Still in the skin.
Still in the skin. And what would the effector be then? Not sweat glands, but
The muscles.
muscles, because
Yeah, you shiver when you're
you
cold.
shiver. Yes. You guys in Canada, you know about this stuff, right?
It was
It's
really cold
cold.
today.
Is it? What temperature is it there?
Um, it's 2 degrees right now.
Two degrees Celsius?
Yeah, Celsius.
Okay, all right.
Fahrenheit, I don't know.
Yeah, yeah. We're one of those backward countries that still uses Fahrenheit. But I can relate to it because I grew up in Holland where they use Celsius. It took me forever to figure Fahrenheit out when I moved back. So then you get the shivering, Patrick, which will eventually get warm because when
Yeah,
the
because movement.
muscles are moving like that, they generate heat, which then warms up the receptors, right?
Mm-hmm.
Okay. So that's sort of one example of negative feedback that works on two sets or extremes of stimulus to bring you to the middle where you need to be. So it's a pretty good example. So let's look at positive feedback. On this one, we're trying to increase the effect of the stimulus. All right, not getting rid of it, but increasing it. So one example would be child labor, which we'll talk about, blood clotting, and why that could be bad as a positive feedback, fruit ripening. All right. So these are three examples that are all very different, but they all involve a positive feedback loop.
Oh wait. Um, for blood clotting, how does it work? Like,
Okay. So you mean how do clots form?
oh, no, we studied that in school actually as part of the immune system. But
Yeah, let's think about this. If I want to have a certain amount of clotting fairly rapidly, especially if I got a decent sized cut, right?
right.
So I want this positive feedback to occur quickly. But what happens if it doesn't shut off? Let's say the clot forms and I'm not bleeding anymore, but I'm starting to form more small clots.
It's dangerous for the body.
Yeah, and there's actually a condition where that occurs where the body itself is so overcome by this positive feedback loop that it develops clots through the entire circulatory system.
Oh, so it's
And
positive feed.
basically you become one big clot, you can't pump any blood and you die.
Okay, so for blood clotting, it's positive feedback because you want the like to increase the effect of blood clotting really fast to stop bleeding.
Yep, exactly.
Okay, I see.
Yep. Okay. Let's look at childbirth, okay, and see what's going on with that. So the big part here is when the baby's ready to be born, usually if everything's right, the head is pointing down, right? And it pushes against that cervix. And there's receptors in the cervix who go, hey, baby wants out, right? And what that then is going to do is it's going to send a signal to the effector in the pituitary gland, which is going to release oxytocin. All right. The oxytocin is going to then give you the response to stimulate contraction of the uterus to push the baby out. Make sense? So when you, this is a lot easier to teach to adults who've been through this because they realize, okay,
Uh,
I'm getting these contractions, but they're not really strong contractions. They're sort of weak, right?
mhm.
And then they start coming regularly, and the docs usually say, hey, when you're getting contractions within this amount of time, like 15 or 20 minutes, they're getting stronger, they're regular, then you need to come in. All right, because it's time for the baby to be delivered.
Yeah,
And
I've watched enough shows to know about this.
yeah, okay, good. So what happens on this positive feedback loop, it isn't one of those things that's immediate. It's like contracting, like, one after another. It is sort of timed, okay? Because if the uterus were to start to contract really fast and the cervix hadn't opened up or dilated enough, then it could literally rip the uterus, which then, mom might bleed to death, right? And the baby would probably die too. So there's some relations in here that aren't on the slide where you get that feedback from the cervix. It's also going, yeah, I'm opening. Give me some time, I'm opening, right? But there is a certain point when it's open enough that this feedback loop sort of goes into fast forward. And it goes, okay, yeah, we're ready. Let's start really pushing and getting uterine contractions that are stronger. And that's because there's going to be more oxytocin delivered into the system. All right. So it's sort of like revving up, and then you're done. But then it takes a while for it to turn off too, because you also want to get rid of the placenta. You have to deliver that too, right? And so you go, hey, your baby's out, we're done. No, you're not done. So this is a positive feedback loop that you need to only know sort of in this simplistic sense, but overall it's much more complex than that. All right.
Mm-hmm.
Okay. So what happens if we have some kind of real imbalance, though? Right. And this is where people like endocrinologists make their money, right? And sometimes these could be caused by things like genetic disorders or drug and alcohol abuse that has caused damage to either sensors, effectors, receptors, or where people become intolerable to things like extreme heat or cold. So, like for me, we've got people trained at hotels when my wife and I travel. I can't sleep unless the hotel room is nice and cold, and so I always have to tell them, look, please set the AC on too cold. Otherwise, it takes me forever to get to sleep, and it's sort of because I have an intolerance to heat. Which means I probably should just go ahead and move to Canada or Iceland or Greenland or somewhere where it's cold. But these conditions, a lot of times, these imbalances are things that are treated by endocrinologists because they involve the endocrine system. All right. So
Okay.
when we think about disease where we can't, do this, maybe cancer. All right. Body can't regulate cell growth, diabetes, can't regulate blood glucose levels. Right. And so for diabetes, you might have to take insulin or metformin, different medicines that help you do that. Of course, with cancer, you might have to do things like chemotherapy or surgery to remove the cancer. We're going to talk about cancer in the second half of when we talk about cell cycle. So I don't want to go too in depth on it, but just realize, let's say, for example, somebody had cancer of the ovary. And of course, the ovaries are going to produce things like estrogen and progesterone. And if those cells are multiplying out of control and they form a big enough tumor, it could cause the woman to overproduce things like estrogen, which, that's not good either, right? So depending on what types of cells that the cancer is in, it can cause over or under production of hormones that then really throw everything out of balance, okay? Because the feedback loops are either overexcited or underexcited, as the case may be. That makes sense?
Yes.
Okay. So if we think about cell signaling and how that relates to homeostasis, then, right, we have to be able to communicate between cells across the body. I mean, from head to toe. And so that what that means then is that that communication goes through what we studied, 2 weeks ago, which was signal transduction pathways. And again, remember we talked about hormones can also cause signal transduction. So when we're maintaining our homeostasis, a lot of times it again a um, it has some kind of link to hormones and therefore the endocrine system. It has sensors and therefore it has a link to the nervous system, right? And so those two systems are really important when it comes to this idea of homeostasis and self signaling.
Wait, which two systems?
When you think about your nervous system for your
Oh,
sensors, right, that actually go, hey, something is out of whack, and signals a organ to release something like a hormone, and therefore also the endocrine system. Those two systems work to maintain homeostasis in the body. Okay. So one of the things, and this, you don't need to do this report, but I do this with my AP kids. I tell them you need to pick two good examples of negative feedback that you know really, really, really well, and two positive ones that you know really, 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 really well. All right. And what you need to know about each of those, it's sort of on this next slide. I make them do a research project and then they have to present it. Okay. And they have to show in a diagram what the feedback loop is with identification of the stimulus, the receptor or sensor, effector, and response. And then what kind of diseases result from that, or what kind of syndromes result from that feedback loop not functioning properly. So what you need to do is sort of pick, you could pick one or two off of each list and then just sort of write it out till you got it. And if you got that, you've got good examples if they ever ask you to give an example of a negative or a positive feedback loop in the body.